Experimentation throughout the early 1900s showed that RNA was the key to protein synthesis. The publication from Nuremberg and Mattaei in 1961 proved the RNA involvement in protein formation and gave us the groundwork that later Nuremberg and Lieder would use to crack the genetic code. mRNA is a type of RNA that carries the code from DNA to be used in ribosomal protein synthesis. The experiment by Nuremberg and Mattaei was set up like so. A colony of E. coli without mRNA was combined with a homopolymer, a long chain of the name base, a radioactive labeled amino acid, and the other 19 amino acids. Translation of proteins was allowed to take place. The resulting protein was checked for radioactivity. Each radioactive amino acid detected was specified by a particular homopolymer. This resulted in the identification of 3 codons: UUU for phenylalanine, CCC for proline, and AAA for lysine. This was a groundbreaking discovery, but more questions arose, like, what is the length of a nucleotide required to make a codon? And which codons coded for specific amino acids? To determine this, Nuremberg and Lieder devised an experiment where tRNA with a radioactive amino acid and ribosomes were filtered through a nitrocellulose filter paper, which allowed the tRNA ribosomes bound with mRNAs to stick to it. The radioactivity of the amino acid indicates if the trinucleotide codon coded for the specific amino acid being tested. This was repeated over and over until the genetic chart was created. This is what geneticists use today to identify genes and patterns in DNA, and is the key to understanding and possibly even mastering the human genome as we know it today.